Effect of epigenetics. Role of DNA methylation is a transcriptional silencing, and it has the effect of tissue-specific gene expression and X inactivation and genomic imprinting. When the fertilized egg cells start to divide in the beginning and generate the stem cells, which can make all the cell in our body, those gene expression pattern is very broad. Almost every gene is expressed, but over the time, when they develop into specific organs, different organ has a different pattern of the DNA methylation, and then some genes are silenced off, and that's why different organs they have a different genetic expression. Even same organ, if you compare those methylation patterns between identical twins, depending on their lifestyle, their the same organ, the methylation pattern also can be changed. So as a result, if you look at the different organs, the methylation pattern, so here the first example is a placenta, which may be similar to the stem cell, which has a much the higher yellow color. That means many genes are expressed. Now they become the blood the cells or different organs. Now you can see chunk by chunk, many genes are silenced. So that is determined by this methylation pattern. So this is unmethylated yellow color. Methylated blue color shows that the general pattern the difference of gene expression depending on the development and also different organs. If you look at human genome, we have 23 pairs of chromosomes. All other chromosomes are identical, but sex chromosome we have differences. If you think maybe the female is the default form of a human because the female's two chromosome structure is very similar to other chromosomes. Male has a very strange one small Y chromosome which has some controlling effect to X chromosome. So female has a 2X chromosome. Male has only one X chromosome and one small controlling part. So if female express both of those X chromosome, now the balance is broken. The difference between male and female will be too big. That's why there is some mechanism that X inactivation. So one of those X chromosome in female is inactivated. That's how we can keep the balance between male and female. If you look at those DNA, and there is a two sides of the X chromosome, one here and one here, and expressed one is only one is expressed. The other one is silent. It doesn't make the RNAs. How do they do it? So this is also through DNA methylation. So the DNAs of this X the chromosome it is highly methylated and it is closed. So in this X activation, seems like out of two X chromosome, through this differentiation, it is dividing and dividing, and it seems like those location or selection of X is looks like a random. So that's why it becomes kind of a mosaic structure. So for example, in cats, the fur color, those genes are located in X chromosome. And then depending on which part of the body they activate which gene and which gene is the inactivated. So it has random color distribution. So this is an example of epigenetic the regulation through X inactivation. Another interesting example is the genomic imprinting. So here is the two genes, A gene and B gene. From sperm, B gene is methylated. From egg, A gene is methylated. So a baby boy was born and it has a mixture of A, B with a methylated and unmethylated. Interestingly, when it make the sperm cells, both of the sperm cells, all of them, B gene is methylated, regardless whether those gene is from father or mother. Well, let's look at the case again. Now, same situation, but if the baby is a female, now they have a same the genetic the expression during its life, but when she makes the egg cell, now the situation, old egg cell, A gene is methylated. So it doesn't matter whether they are from father or mother. So it seems like there is some mechanism that female prepares one gene, in this case B, that's why it methylates A. Male prefers A gene because it methylates B. So this is so-called genomic imprinting. Now, DNA methylation change is it seems like when the egg cell and sperm cell meet together, still there is methylation pattern, but very early stage of the pregnancy, most of those the methylation pattern is deleted. And then from the blastocyst stage to make embryo, the new methylation pattern is restored. So genetic imprinting implies that there is some genes, they survive here and then original methylation pattern in sperm and egg, they remained, not fully deleted here, they remained and provided the impact to the embryo. Do you know this person, Audrey Hepburn? She was a very famous actress and she was a pretty and lean body and even though she eats food, her body did not get fat. So people thought maybe that is her blessing. But her symptom is from sad history. During the World War II, there was a serious food supply problem in Netherlands. Many people, including her, suffered from the famine situation for many months. So after war is over, even though everything becomes normal, her body has a long-term memory about this, and it seems like epigenetic changes made a very abnormal situation to her body. It seems the effect of this famine is more significant during the pregnancy. If the famine happens during the late stage of pregnancy, baby is born as a small size, but in general, the baby is okay in a later stage, but with some minor diabetes cases. The effect was much more significant in early stage, and if it happened in before the 3 months of pregnancy, the baby it looks like the normal size when they were born, but later they develop obesity, hyperlipidemia, or cardiovascular disease, many more problems. This may be because the methylation pattern is made in an early stage of embryogenesis, and this is uh, the clear the evidence that epigenetic modification keep long-term history. Even interestingly, but sadly, this effect not only ends at the baby's the life, this gene's effect can be transferred even to the offsprings. So that means there are some cases that epigenetic changes, not the genetic sequence change, only methylation changes, it can also can be inherited to the next generations.